Thank you for the kind introduction. Today I'm going to talk about the effects of HIV on aging of the brain. It's been known since the early days of the HIV pandemic that older individuals have more difficulty with cognitive function. And as shown here from work done by the CDC in the early 90s and late 80s, that those individuals at greater than 60 years of age have a rapid decline in cognitive function. And now, even in the antiretroviral era, whereby frank dementia is not that often seen, greater than 30% of individuals, even if their CD4 cell counts are under decent control, they still have cognitive difficulties. And just adding more antiretroviral drugs does not seem to solve the problem. And several studies confirm this now, and it's shown here on the left. You can see that even individuals who are less than 60 years of age have a more rapid decline in cognitive function. And here you can again see that older individuals who have HIV infection have more complaints with regards to their activities of daily living. And if you look at the type of dysfunction in these patients, you will see that these patients on antiretroviral drugs more often have problems with executive function, as you can see here. And they have more difficulty with learning. And so it's not all cognitive domains, but some domains are much more affected than others. One might argue that what kinds of effects that really has on a patient's functioning and long-term survival, and it's shown here that just by the fact that those individuals who have cognitive dysfunction tend to die faster. And that is because if you can't remember to take your medications, and if your cognitive function is impaired, you can't protect yourself in many different ways. And so a variety of different reasons can contribute to early demise in these patients. We looked at patients by age, categorized them into normal controls, those with asymptomatic impairment, mild, and those with frank dementia, and looked at neurofilament levels in these patients. And what we found was that the neurofilament levels go up with age. And also protein carbonyls, which is a sign of oxidative stress, also seem to increase with age in this patient population. And so, and these are individuals with HIV infection. However, the PET scan studies have not been all that conclusive. It's been hard to demonstrate a clear difference in the HIV positive patients compared to controls for increased uptake of the marker that binds to amyloid, showing that there's increased amyloid deposition. However, in some areas that might be the case. For example, here in the cingulate gyrus here, and you can see in the prefrontal area, there seems to be more deposition of amyloid than in other areas of the brain. Pathological studies consistently show that patients with HIV infection have more deposition of amyloid plaques. And so you can see that even at this age of 30 to 39 age group, these patients with HIV infection had more amyloid plaques in the brain compared to healthy controls. And at every age group, that seems to be the case. And here in another paper, they've shown the shape of the plaque. And you can see here, they call them diffuse plaques. So in HIV-infected individuals, you can see these plaques, but the borders of them seem to be a bit diffuse. And while in Alzheimer's plaque, you can see very crisp borders over here. But if you quantitate these number of plaques, you will see that they keep going up with the length of infection. Then how does HIV really lead to these plaque formation? And one way is to try to understand where HIV itself is being produced in the brain, and then what the mechanisms might be for interaction between the two. So HIV is a complicated genome, and it has, it's a retrovirus, so it gets integrated into the DNA, and then a number of different genes are formed by splicing of the RNA. And so one of the first proteins to be formed is TAT, and it's formed off of 2 exons. And so if you look at, by immunostaining in the brain, you'll find that TAT is present here in the perivascular cells, and these are all macrophages here, all this brown staining with it. And you can also see it in these microglial nodules. Okay. Here is staining for GP120, which is the envelope protein. And you can see these multinuclear giant cells staining brown, showing that there's increased production of HIV in these cells. You can also find HIV in astrocytes. And here you can see immunostaining for the NEF protein, and in situ hybridization showing the RNA for the GAG pol transcript in astrocytes. However, even though HIV is present in astrocytes and in macrophages, the cells that degenerate are the neurons, even though the neurons themselves are not infected. So you can see here that in the dentate gyrus, this is what the normal dentate gyrus should look like. This is a patient with no encephalitis. This is one with drug abuse. You can see that there's decreased staining. And if you look at this photomicrograph here, this is what the normal neuronal population should look like. And an HIV patient with dementia here, you can see there's profound loss of neuronal staining. Now, if you were to take a transgenic mouse in which there is made with these 2 genes, amyloid precursor protein and presenilin-1, they are at increased propensity to develop amyloid deposition, and cross them with the GP120 mouse. You will find that in these crossed animals, there are these huge plaques that form here of amyloid deposition. And you can quantify them, and you can see that in different regions of the brain, starting from the bregma and going posteriorly, you will see that there are huge amounts of plaques in the triple transgenic animals. And the size of the plaques is also greater in these animals. So the number of plaques as well as the size of the plaques. And now I'm going to show you that not just only GP120, but even the TAT protein can interact with amyloid as well in a very unique manner. So the TAT protein, as I mentioned earlier, can be found in these perivascular cells, but is also present as deposits in the extracellular space here. And it tends to aggregate very easily. So you can find these aggregates of TAT protein in this microglial nodule here. And you can also find TAT in the spinal fluid of patients who have been adequately treated with antiretroviral drugs for a long period of time. So this is an ELISA for TAT, and you can see that there is increased TAT that can be found in the spinal fluid here. You can also find it in exosomes from the spinal fluid. And the presence of TAT has consequences because those individuals who have TAT detectable in the spinal fluid also had difficulty with information processing, and psychomotor domain was also diminished in these patients. And interestingly, we found that there was a propensity to be present more so in women and individuals who are actually even better controlled on antiretroviral drugs than others. And so it shows that just control and recovery of CD4 cell count alone is not sufficient to prevent its production. And that there's an inverse correlation in the presence of TAT RNA and HIV RNA and TAT, suggesting that once the virus is controlled, the proviral DNA is probably still trying to crank out TAT and because it's so critical for HIV replication. So there is this positive feedback loop that leads to increased production of TAT in the presence of antiretroviral drugs. But it has multiple consequences. TAT can act on T cells and it can activate T cells. So you find a lot of activated T cells in these patients. The other thing it can do is it can directly interact with neurons and cause excitotoxicity. So as shown here, you can see that when you put TAT on these hippocampal slices here, they just start firing. And you can see that each line here is an firing from these neurons, and it just fires like crazy all the way through. If it acts on glial cells, it can lead to production of cytokines. So it can activate glial cells, and you can see IL-1, IL-6 being increased in a dose-dependent manner. And it also synergizes with other viral proteins, for example, with GP120 or glutamate, in order to cause more excitotoxicity. So here's a cell that is repeatedly treated with glutamate, and you can see the amount of calcium changes inside the cell decrease. However, if you pre-treat the cell with TAT and now put glutamate on there, you will see that the cell can no longer handle the calcium, and there's huge increases in calcium here. Similarly here, with subthreshold levels of GP120 and TAT put together, you see increased levels of calcium within the cell. And in transgenic mice here, if you put TAT and APP together, you'll see that there are these huge plaques that will form of amyloid beta peptide plaques here. And you will see that if you put them in culture, you will also notice that the neurons themselves start degenerating. So here's amyloid beta peptide with TAT, and we counted the number of puncta, which is an indication of neurotoxicity, and they go up in a dose-dependent manner. And if you look at the interactions of TAT and amyloid beta peptide by microscopy, what you will see is that here's the amyloid fiber, and these little bumps on top are the TAT sitting on there. So they decorate the fiber really nicely in a systematic manner, and ultimately they form lots of different types of plaques. And that's shown here diagrammatically. What TAT will do is just wrap these amyloid plaques together, and ultimately they form these huge plaques here. And these will then sit on a neuron and lead to a toxicity of the neuron by making pores in the cell membrane. If you were to take TAT and put it in different regions of the hippocampus, you'll find that there's some parts that are more vulnerable than others. So the dentate gyrus here and the CA3 region are much more susceptible than other parts of the brain. So the question is now, can we block the effects of TAT? And the answer is there are many different ways of being able to do it, although none of them are currently therapeutically available. So this is beautiful work done by Susanna Valenti, and she's shown in a series of papers that this drug called cortistatin A can inhibit TAT, but once it does so, it will just lock HIV completely. So no other transcripts will be formed. So that's called a block and lock type of phenomenon. And because TAT is also responsible for chromosomal rearrangement, so once it's the chromosome is open and TAT is available, if you were to be able to block it, then it'll just lock the entire HIV genome right there for extended periods of time. And in our lab, we have made antisense molecules against TAT in order to be able to prevent the protein from being formed. And so these antisense molecules are strands of modified DNA and a combination of DNA and RNA moieties here, and they will then bind to the messenger RNA and lock it and prevent it from forming a protein at all. And we've shown here in these astrocytes where TAT is being expressed, and these are a panel of different types of antisense molecules, and we've identified some that act really well compared to others. So in summary, there is an acceleration of neurocognitive deficits with aging. There's uncontrolled TAT production and deposition of amyloid in the brain, and despite use of antiretroviral drugs. And a lock and block strategy with TAT antagonists can potentially provide a functional cure and stop age-related progression. So I'd like to acknowledge a number of people who have contributed to this. A number of them are not on this list, and I apologize for that. But people who developed the TAT assay was Carol Anderson, Muzna, and then Tori Johnson in my lab. All the work with TAT amyloid interactions was done predominantly by Alina Hadigan, and the therapeutics work is being done by Lisa Henderson, who is leading the efforts. Thank you.